Hi, my name is Cole Malloy, and I'm presenting the effects of potassium concentration on resting membrane potential and its role in enhancing deep tissue injury. The goals for this procedure are to understand the ionic basis for resting membrane potential, to learn the methods for measuring membrane potential in the crayfish extensor muscle, and most importantly, to study the effects of deep tissue muscle injury, or DTI, and the influences on surrounding healthy tissue and neuronal function due in large part to increase in extracellular potassium following injury. This analysis has obvious relevance to human health. DTI results in muscle tissue damage, which induces cell breakdown and release of intracellular contents. These contents, which include ions such as potassium, and the effects of this ion release on surrounding cells has not really been studied extensively. Studies have shown also that a rise in extracellular potassium ions from activity of cells can influence the resting membrane potential in neighboring cells that were not initially electrically active, and could result in depolarization of these surrounding cells. In addition, we know that depolarization can result in recruitment of voltage-gated ion channels to open and potential sodium channel inactivation, which obviously can impact synaptic transmission. In addition, consideration in the exposure time and the effects of restoring normal potassium concentration on the health of skeletal muscles and synaptic transmission hasn't really been fully addressed. Here we see a couple of illustrations of DTI. As you can see, especially in the illustration on the right, the muscle atrophy spreads over a rather large area, protruding away from the original injury site, suggesting that surrounding healthy cells are affected by release, perhaps, of the intracellular contents from the damaged cells. Thus, the data gathered in this procedure will help to answer the questions relating to the aim of this project, which is to investigate the effect of the release of intracellular ions, like potassium, on surrounding originally undamaged cells. So how does damaging cells as a result of deep tissue injury affect surrounding healthy cells? So I'll quickly go through a quick review of the ionic basis of resting membrane potential. We know that an ionic gradient across a biological membrane can result in the membrane potential. The two main factors affecting membrane potential are ion concentration on either side of the membrane, as well as the ionic permeability of the membrane. The quantities and movements of different ions, such as potassium, sodium, and chloride, together determine the membrane potential. Here's a schematic of a cell at rest. Although concentrations of ions on the inside of the cell vary with cell type, in general there's a high intracellular potassium concentration and low chloride and sodium concentrations in relation to the outside. At rest, the potential inside the cell is around -71 or -70 millivolts relative to the outside, and this is mainly due to the flow of sodium and potassium ions across the membrane, which is regulated by the sodium-potassium ATPase. This ATPase pumps 3 sodium ions out of the cell and 2 potassium ions into the cell. When the membrane is at rest, it is generally more permeable to potassium, and this is due to the presence of potassium leak channels. Thus, because of this, the membrane potential is closer to the potassium equilibrium potential than it is to the sodium equilibrium potential. So the main question addressed in this analysis is, what happens to the resting membrane potential when extracellular potassium is increased? We can relate this to increased potassium concentration from intracellular stores of muscle cells that are damaged as a result of deep tissue injuries. Secondly, to what degree does this affect the efflux through potassium leak channels? So here's a schematic of what we're thinking about. So again, injured cells leak potassium ions, and these potassium ions can have a dramatic effect on the resting membrane potential of neighboring cells. We're looking to find out how this leakage of potassium ions actually affects the resting potential in neighboring cells. Here's the experimental setup that was used in this procedure. A glass microelectrode was used to measure intracellular cordings from the crayfish extensor muscle, which was exposed via dissection and placed on a Silgard dish. Various solutions were washed onto the prep to examine the change in the resting membrane potential upon altering the potassium concentration in the solution. The data was then recorded with Lab Chart 7, which depicted and illustrated the changes in resting membrane potential. And here's a closer look at the dissected prep with the glass microelectrode tip and the extensor muscle. The crayfish is ideal for electrophysiological studies, such as this one, for a number of reasons. Number one, the dissection is relatively simple, and the dissection allows for the exposure of muscle, which is relatively easy. In addition, the crayfish prep is viable for a long period of time in a physiological saline, and the potential differences across the membrane can be easily recorded. On the right is an illustration of the internal anatomy of a crayfish, and the extensor muscles, which was used for this analysis, is illustrated. Here is the preliminary data collected from our group. The graph on the right depicts the average percent change that was calculated for each exchange from saline to a solution with varying concentration of potassium. As is illustrated, the percent change in resting potential increased as the extracellular potassium concentration in the solution was increased. The table on the right lists the potassium concentration in the saline, which was 5.4 millimolar, as well as the potassium concentration in the muscle juice that was used. The muscle juice consists of 1 part homogenized pure crayfish muscle and 3 parts saline. And it should be noted that the homogenized crayfish muscle, or the concentration of potassium in the pure homogenized crayfish muscle, is approximately 130 millimolar, and this was found by Bernard Katz in his analysis way back in 1975. So upon dilution, the potassium concentration in the muscle juice used was calculated to be 36.55 millimolar. As can be seen by the graph on the left, there was a positive percent change of about 24% when the saline was swapped for muscle juice plus saline. And as the potassium concentration increased in the solution, so too did the average percent change in resting potential. Here's an illustration of the raw changes in resting membrane potential for each group. As you can see, increasing the extracellular potassium concentration to 40 millimolar from 5.4 millimolar saline resulted in an increase of between about minus 65 to minus 37 millivolts. It's noted that a more dramatic increase in RP was observed when a 60 millimolar potassium concentration solution was bathed on the preparation. So this is an exchange from 5.4 millimolar saline or 5.4 millimolar potassium to 60 millimolar potassium. The RP upon exchange from 5.4 to 60 increased from between minus 43 millivolts to minus 31 millivolts for our group. So a dramatic increase in RP was observed after increasing the concentration of potassium to 60 millimolar. This graph depicts a linear change in RP examined by our groups as the extracellular potassium concentration was increased in the solution. There was a positive linear change in resting membrane potential as the potassium concentration was increased, as can be seen in the graph. In addition, a line was calculated for the hypothetical values that should have been hypothetically calculated based on the Nernst equation. It appears that our groups underestimated the positive RP values that should have been calculated based on our analysis, as the hypothetical values suggested that the resting membrane potential was actually more positive than what was found with our groups. However, each displays a positive linear increase in resting potential as the potassium concentration is increased. If we think about DTI and the effects of surrounding cells upon release of potassium and how it relates to human injuries, we can think about the actual concentration that was calculated in crayfish muscle, intracellular potassium concentration in crayfish muscle, which was found, as I presented, to be 130 millimolar. Suppose that this concentration is close to the intracellular potassium concentration in human muscle cells, and what the effects would be in dumping a concentration that high, how that would affect surrounding cells. One would imagine that it would be rather dramatic. So we now know that increasing potassium concentration alters resting membrane potential, but what happens also when sodium and other intracellular components are released, that is to be determined. We know that potassium obviously is not the only component that's released, so further examination is needed in this regard. In addition, what does intermediate and long-term exposure to high concentrations of potassium do to synaptic transmission? Well, analysis by Dr. Cooper and his colleagues have shown that in a 1-to-1 dilution of muscle juice with saline, no evoked responses were measured when a sensory to motor circuit was stimulated. This suggests that synaptic transmission is severely disrupted upon exposure to a concentrated muscle juice solution. And it can be noted that the RP here was measured to be minus 50 millivolts, which our groups recorded in our analysis. Thus, it appears that increasing the extracellular potassium concentration has detrimental effects on synaptic transmission, and these studies can help to provide evidence that DTI could affect healthy cells due to an increase in the extracellular potassium concentration as a result from the dumping of intracellular stores after damage. So in the future, this study can aid in helping target new therapeutic methods for treating DTI, and our procedure helps to provide preliminary data to back this proposed analysis.